Good afternoon, everyone. We all know that there's a lot of hype surrounding the promise of pharmacogenomics and its possible impact on personalized medicine. And I think part of the reason that there's emphasis on the topic we have today on evidence generation is that I think we can all agree that there needs to be evidence backing up introducing testing for adjusting prescribing of medications, and we want to try to temper the hype where necessary. Several of you have pointed out to me that I have finally hit the big time because my name, along with several others in this room, is mentioned in the Delta Sky magazine. So those of you who are flying home can read about personalized prescriptions and pharmacogenomics if you're taking Delta. And despite this hype and enthusiasm that has reached the airline industry, the use of pharmacogenetically guided prescribing is still not widespread. So I like this study that was published last year in CPMT, a large academic medical center at University of Washington with over 130,000 patients, over 3.2 million clinic visits. Hundreds of thousands of medication orders were written for medications that have germline pharmacogenetic testing mentioned in their FDA labeling, 49 of which are actionable by CPIC, and only 1.5% of prescriptions for those drugs where testing was either recommended or required by the FDA actually were accompanied by testing. So it's still true that it's a very, very small minority of prescriptions that could benefit from pharmacogenetic testing actually have pharmacogenetic testing. And when we surveyed experts now some time ago to ask their top 3 challenges as to why they weren't implementing pharmacogenetics in the clinic, 95% of respondents said that the process required to translate genetic information into clinical actions was a major impediment. And the next 2 responses were how to interpret genotypes and which gene-drug pairs should be implemented first. And we repeated this last year and got very similar results. So back in 2011, Dr. Terry Klein of Stanford and I started the Clinical Pharmacogenetics Implementation Consortium, and our goal was to create gene-drug pair guidelines that were freely available, updatable, and evidence-based that could be used by the community for helping with that process of translating genotype into prescribing actionability. Our goal is to provide these guidelines. I don't have enough room for the mouse here. And it's a really important assumption underlying these guidelines that we are not stating whether genetic tests should be used. We are stating, pretend that you've all had your genomes done. We've just heard how many people have had their 23andMe's already done. The challenge for clinicians is going to be deciding which genetic variability is so strongly associated with drug prescribing that it would be wrong not to use it if you had it. And thereby we avoid much of the evidence that's required in medicine related to things like cost effectiveness. And we're not having to prove that genetic testing is cost effective than not genetic testing because the patient is already there with their genetic tests in hand. We're saying pretend you have the genetic test results, how should you use it? But nevertheless, of course, we look carefully at evidence. And we have an algorithm for how we decide prescribing actionability level to gene-drug pairs. And we try to grade gene-drug pairs as level A, B, C, or D. I don't have time to go through all of these different pieces of evidence that are used in deciding how to tentatively assign the actionability for gene-drug pairs. But CPIC level A or B, there is a prescribing action recommended of some sort, and examples here would be mercaptopurine and warfarin. A CPIC level C is we can't make a prescribing recommendation based on this gene-drug pair. Either there's not enough evidence or the alternatives are unclear. But it's important to assign these grades because testing for that gene might be important, or marketing for that gene-drug pair might be taking place by the private sector, and we need to provide clinicians with guidance on what to do with those gene-drug pairs. And then CPIC level D is, of course, the majority of drugs where there's nowhere close to actionable, clinically actionable evidence. And the other part I want to emphasize is that CPIC is not just looking at the evidence that links the gene to the drug of interest. We're looking at the evidence for the alternative therapy. And if the evidence for the alternative therapy isn't strong enough, then we're going to have trouble making a prescribing recommendation. There's about 120 drugs that we've classified as CPIC level A or B. This is about 10% of approved drugs in the United States. The page where we list our assignment of the actionability to these gene-drug pairs is very heavily visited. We had over 36,000 hits to this page last year. And you may visit this page and argue with our tentative level, and we'll be happy to consider changes. But obviously, we really don't know the level to assign to these gene-drug pairs until we get into the business of trying to create a guideline, which is very labor-intensive. So in general, when do we use a test to guide prescribing? Well, we use it when the drug has a narrow therapeutic index for safety reasons. The difference between the dose of the drug that that can cause that causes the intended therapeutic effect is not much lower than the doses that can cause serious adverse effects like anti-cancer drugs. Or where the drug has unpredictable non-dose related adverse effects, such as we'll hear about tomorrow from Dr. Phillips on Stevens-Johnson syndrome. Where, of course, the effective treatment of the disease is important, cancer, seizure control. Most of us don't want to admit that maybe the treatment of our disease isn't the most important thing in the world, but probably whether I have a really good sleeping pill for my trip across the Atlantic to Paris isn't as important as whether a child with cancer is cured of their disease or not. Where the pharmacodynamic effects are not readily titratable, so that also applies often to cancer, where we don't know till years later if we've successfully treated the cancer, whereas for something like blood pressure, you may have an intermediate, quickly available monitor of drug effect, or where the drug has some advantages over drugs with a low genetic risk. So that applies, we think, to drugs like codeine over morphine. In pediatrics, we would rather use ondansetron over granisetron for formulation reasons. CPIC publishes its guidelines, usually in clinical pharmacology and therapeutics, and a few examples are listed here. And they are also simultaneously posted on the website, so every guideline, gene-drug pair, has its own page on the CPIC website. And we try to emphasize to people it's very important to go to the CPIC website because that's where we put updates. And we do add updates since the publication, adding gene variants or other new prescribing information until such time as we can fully update the published guideline. So these evidence-based guidelines are highly used. We collaborated with NHGRI and surveyed sites that were doing clinical implementation of pharmacogenetic testing last year. And we found that 34 of 36 sites that are doing clinical implementation of pharmacogenetics were using CPIC guidelines as part of their resources. Every guideline, we have a detailed SOP for how we write these guidelines. Our volunteer authors are often not very crazy about these detailed elements of the SOP, but it's important because the evidence review is really a critical part of these guidelines, and I'll talk a little bit more detail about the evidence review. So the heart of the CPIC guideline is the prescribing recommendation. How do we link phenotype to specific prescribing recommendations? And we grade those prescribing recommendations as strong, moderate, or optional. This is based on a system that was used by the HIV group. And for a long time, we tried to avoid having no recommendation, because obviously having no recommendation is not very useful to clinicians. But what we have found is that there are some gene-drug pairs where one phenotype or one drug might have a moderate or strong recommendation, and another phenotype for that same gene and that same drug, there may not be enough evidence to make any recommendations. So we do have that as a category available to us. And an example here is for CYP2D6 and ondansetron tropizatron, where we have a moderate and strong recommendation for the ultra-rapid and normal metabolizer phenotypes, but we were not able to make any recommendation based on the intermediate and poor metabolizer phenotypes, despite really looking for it. We couldn't manufacture one where the evidence just didn't support it. So we have to grade the evidence, and part of what we do is grade evidence that assigns function to alleles. This is a critical part. It's gaining more attention. We have to beef up this part of our evidence grading, but it is part of every CPIC guideline. And we use the evidence the evidence that's used to support the prescribing recommendations is what takes the most time. Of course, it's based only on published peer-reviewed literature. So there are some European and FDA drug labels that have pharmacogenetics incorporated without published data to back up those recommendations. And I'm not saying that the data aren't real, but we've decided that we can't evaluate something that's not published. We have standardized inclusion and exclusion criteria for what we evaluate in the literature, and we have working tables while the authors are going through all of the original research that supports the gene-drug pair, and we score every major finding as high, moderate, or weak. And we do this with all of the authors on the guideline. Do the studies support or not support the finding? And if needed, we go down to the level of scoring individual studies as to their quality and flaws. And we borrow heavily from a system that's been used by the ASCO guidelines for cancer guidelines. Overall, for scoring evidence, we say that the evidence from a study is high if it's consistent from a finding, is consistent across well-designed, well-conducted studies, moderate if there is evidence that's sufficient to determine effects, but the strength is limited by the number, quality, or consistency of the studies, and weak if the evidence is insufficient to establish the effects on health outcomes, et cetera. So some of the types of evidence that we look at in support of a gene-drug pair include in vitro functional studies of the gene variant in the drug, case reports, preclinical and clinical studies that link pharmacologic effects or drug concentrations to genomic variation, in vivo PK, non-randomized clinical studies, often retrospective studies that may be very strong, associating genetic variation with a drug effect. And then, of course, the gold standard is randomized clinical studies of a genetically based prescribing arm versus a standard of care arm. But these are almost never done in pharmacogenetics because by definition, the most important variants are rare and the sample sizes are often too small to conduct such studies, or it's considered unethical to avoid the genetically based prescribing change in that small percentage that have the high-risk phenotype. I'll just point out that it's very helpful to us to use the PharmGKB system for clinical annotations. They've been evaluating the literature. I saw Michelle earlier for, I think, about 20 years, and grading findings for gene-drug pair assignments using these clinical annotations, and we utilize that information as part of the CPIC evaluation whenever possible. I went through this recently because Dr. Jun Yang and I got to update the thiopurines guideline, and I was able to see for myself how this works and how we drive the PharmGKB curators crazy. I know this is hard to read, but for example, here's a finding that the TPMT variant is not associated with a greater likelihood of event-free survival. Here's a finding that TPMT status is associated with development of second cancer. This is one of my original findings, but even I gave this as weak evidence in the overall consideration of the finding of relating TPMT genotype to second cancers because there were many conflicting studies that did not find the same thing. So every important piece of evidence that goes into deciding how are we going to adjust prescribing of thiopurines based on TPMT variants is graded by the authors of the guideline. So the other thing I mentioned is it's really important to look at the evidence related to the alternative therapy that doesn't have anything to do with the gene and drug of interest. Again, of course, the therapeutic index for the drugs involved, the severity of the underlying disease, the consequences of suboptimal prescribing, the availability of genetic tests, the availability of an evidence for that alternative therapy. So one example is clopidogrel. We heard about this earlier from Alan. It requires CYP2C19 activation in at least 2 different steps to form its active metabolite. And so biochemically, that's been known for some time that that was the case. Alan showed in a GWAS that in fact it was CYP2C19 that was the primary hit, where the phenotype was platelet aggregation response to clopidogrel. And there were some beautiful retrospective studies showing that patients were at a much higher risk of experiencing an adverse cardiovascular event if they, depending on the number of these poor metabolizer CYP2C19 alleles that they carried. And this was even stronger, of course, if they were taking the drug at the time of the event. And subsequently, there has been a randomized trial, or a trial at least showing the effect of these in real time, the effect of cardiovascular adverse events in patients who have loss of function alleles who continue to get clopidogrel despite the presence of those loss of function alleles, in the patients who didn't have any loss of function alleles for CYP2C19, or in the patients who had loss of function alleles but were given the alternative therapy. And so these data really help to give confidence in the CPIC guidelines, recommendations where there's an algorithm by which alternative therapies such as prasugrel, or however you say this one, is recommended for intermediate and poor metabolizers of CYP2C19. So this evidence was really used in the prescribing recommendations for this gene-drug pair. So as I mentioned, the extra gene-drug pair evidence is really important too. So here we have in clopidogrel, the therapeutic index of the drug is low, and we're looking at the risk of bleeding in the case of overtreatment versus thrombosis in the case of undertreatment, the severity of the underlying disease, stroke or myocardial infarction. The consequences of suboptimal prescribing are very serious, death or serious morbidity. There is availability of genetic tests in the clinic, and there is some evidence for the alternative therapy. So this meets all of those criteria of why we would consider this evidence to be adequate for acting on CYP2C19 genotype if you knew it. So part of our confidence in deciding on how to act on gene-drug pairs has to do with the mechanisms by which the genomic variants are affecting the phenotype. So we can think of the most worrisome kind of evidence being intergenic variants, perhaps affecting enhancers or unknown genomic variants. The association with phenotype might be via unclear mechanisms, and an example of this is this locus on 12q associated with hydrochlorothiazide response. Then, of course, there could be non-coding, coding, cis-regulatory or structural variants via a gene that doesn't affect drug pharmacokinetics, but might still have a very strong association, such as an HLA variant associating with adverse drug reactions. And then the easiest type of genetic variation to understand, coding, cis-regulatory or structural genomic variants that might affect the drug's pharmacokinetics. And the phenotype association is via pharmacokinetics. And what we just saw with CYP2C19 and clopidogrel response is such a mechanism underlying the pharmacogenetic association. And I would argue that our need for sophisticated and expensive clinical evidence to support acting upon that gene variant decreases in this latter type of pharmacogenomic association, and is obviously higher when the mechanism is more unclear. David Veenstra and I wrote about this for the IOM a couple of years ago, and wrote that prescribing decisions are routinely made on the basis of imperfect evidence and extrapolations between solid evidence of mechanisms underlying interpatient variability in drug response and unstudied clinical scenarios. So, for example, dosing based on liver dysfunction, on renal dysfunction, on trying to avoid drug interactions, on almost all prescribing decisions that we have to make in pediatrics is based on extrapolation from other data in other populations and not based on clinical trials in those specific populations. If we look at the CPIC guidelines that exist right now, where there's 18 genes and 30-something drugs, I think, affected, you can see this list here. And if we color code those drug gene pairs that are associated with each other because of pharmacokinetic mechanisms, about 55% of these actionable genes are affected via pharmacokinetics. That means and about 77% of the actionable drugs are affected by their pharmacokinetic association with the gene. Now, of course, this doesn't take into account the fact that some of these drugs are very widely prescribed and some of them are rarely prescribed. So another example of such a pharmacokinetic association is the CYP2D6 activation of codeine to morphine, which of course does not take place in poor metabolizers. So here we see that when we give codeine to morphine in the 90% of the population that are genetically extensive metabolizers, they're both effective analgesics and placebo doesn't do anything. But when we give codeine to poor metabolizers, it is the same as placebo. And this is strictly a pharmacokinetic problem. You can't make the active metabolite of codeine if you don't have CYP2D6. So that means that about 12% of the population should not take codeine based on CYP2D6 genotype alone, 10% because they're poor metabolizers, 2% because they're ultra-rapid and form high levels of the active morphine metabolite. That's a drug where there's an example where there's a prodrug and an active metabolite. Voriconazole is the active drug. It's an azole antifungal. Luckily, we have therapeutic drug monitoring, and the plasma levels of voriconazole are directly related to the CYP2C19 genotype of the patient, which those patients having two inactivating alleles for CYP2C19 have high levels of voriconazole, and the ultra-rapid metabolizers have low levels of voriconazole. So when we want to make a drug adjustment based on these gene-drug pairs where we're really extrapolating from pharmacokinetics, we can take those data into account in deciding how to act on that. And we consider adjusting based on pharmacogenetics completely analogous to adjusting the dose on something like creatinine clearance, reflecting renal function for acyclovir, bilirubin to adjust anthracycline doses based on liver function, and using weight and body surface area to adjust doses of drugs for children compared to adults. And if we insisted that we had a randomized clinical trial comparing standardized dosing for acyclovir to reduced dosing based on creatinine clearance for every level of creatinine clearance, that would be an unethical and waste of precious clinical resources. And you can see that the dosing guidelines in the package insert for a drug like acyclovir are very specific for different levels of creatinine clearance, percents of recommended doses, and dosing intervals, with no randomized prospective trials to back up such pharmacokinetically based recommendations. And a similar type of table is listed here for daunorubicin and the level of serum bilirubin. And I'm not arguing that it's unethical to use pharmacokinetic principles to make such dosage adjustments based on liver function, renal function, and age. I'm arguing that we should treat pharmacogenetic laboratory test results more like these types of test results than as if we didn't understand the mechanism. So our need for clinical trials that compare dosing with and without genetically informed prescribing decreases as we know that there's a pharmacokinetic basis for the drug-gene relationship, and that affects the level of evidence that we require for such gene-drug pairs. Now, 45% of currently actionable genes are not pharmacokinetic, and that affects about 33% of the actionable drugs, and they're highlighted here in purple. And we may need higher levels of evidence to implement such pharmacogene drug pairs. We may need randomized trials in some cases, but even those might not be needed if there is a solid mechanism. So, for example, understanding that variants in the CFTR affect response to a cystic fibrosis targeted drug might not require large randomized trials to convince ourselves that that could be the basis. We know that G6PD has a huge impact on affected drugs and also may not need randomized trials for these associations. Nevertheless, we have this beautiful randomized trial that was done for HLA genotyping related to allergic reactions to abacavir, done by Dr. Phillips and Malal, showing that there were fewer allergic reactions in the patients that had prospective genotypic screening than those who did not, and this became widely accepted in the HIV community. So how do we decide if the evidence is high enough to implement in the clinic? And I'll use NUDT15 as a use case. So based on 30 years of candidate gene studies, started by Dr. Dick Weinchelbaum, who's here in the audience, we and others have acted upon gene variations in TPMT to guide thiopurine dosing for some time. It was time to update the CPIC guideline, and so we really wanted to evaluate whether it was time to include a new gene, NUDT15, in this guideline. And Dr. Jun Yang at St. Jude had showed, because he studied a population of patients that were included many Asians for the first time, along with other individuals of diverse ancestry, and our GWAS of dose intensity and dose intolerance for thiopurines, TPMT was still one of the top hits, but there was this additional hit in the gene NUDT15. And retrospectively, you can show that the dose intensity that is arrived at in patients that they can tolerate is lowest in those who are homozygous for inactivating variants in TPMT, intermediate in heterozygotes, and highest in those who are wild type. And a very similar level of dose intensity breakout by genotype was achieved when patients were divided into their NUDT15 inactivating variants. And we know that NUDT15 activates the triphosphate active thioguanine metabolites of thiopurine, that it dephosphorylates those active metabolites, and therefore is an inactivating enzyme for these drugs, so that those patients who have NUDT15 variants have higher levels of TG incorporation into their DNA, more DNA damage, and more cytotoxicity from doses of thiopurines. And then June went on to make the knockout mouse for NUDT15 and could show that at fixed doses, here at 20 or here at 1 milligram per kilogram, that the wild type mice had much higher ANC than the knockout mice did at either one of those doses, and that corresponded with the level of DNA incorporation, TG incorporation into DNA, where the knockouts had much higher levels of incorporation than the wild type mice did. And interestingly, you could show that you got about the same level of neutropenia and about the same level of incorporation into DNA at a dose of 1 milligram per kilogram to the knockout and 20 milligrams per kilogram to the wild type mice. So is that enough evidence to implement NUDT15 in the clinic? Well, let's just review quickly the evidence that we have for TPMT. It's a life-threatening disease, leukemia with life-threatening toxicities of the drug. Retrospectively, we have solid mechanistic data. Thiopurine metabolites are associated with outcome and toxicity. We have an intermediate. We can measure TPMT enzyme activity in red cells, and genotype can be used to predict enzyme activity, outcome, toxicity, and thiopurine metabolites. We have data that decreasing the dose in those who have TPMT defects maintains high levels of active TGN metabolites, and dosage changes based on TPMT avoid toxicity and did not compromise anti-leukemic control. Dose titrations were accepted for this, and without using TPMT genotype, because of the multi-agent nature of treating cancer, you'll titrate the wrong drug if you don't know that the problem lies in TPMT. So what about for NUDT15? It's a life-threatening disease with life-threatening toxicities from the drug, and retrospectively we have solid mechanistic data. The genotype relates to TGN incorporation into DNA and toxicity. We know that decreasing the dose in those with NUDT15 defects likely achieves their desired level of myelosuppression in retrospective studies. Unfortunately, we don't yet have an easy way of measuring the NUDT15 enzyme activity as an intermediate metabolite, But again, we accept dosage titrations, and therefore we have incorporated NUDT15 along with TPMT. So now we use 2 genes to adjust the dose of starting dose of thiopurines in patients with leukemia. We're doing this at St. Jude in our Total 17 study, and June has been working with the Children's Oncology Group to try to get this incorporated into the next suite of ALL studies there. So just to bring it all back, let's remind ourselves that there's about 120 drugs that are CPIC level A or B genes. It's about 10% of drugs. We don't think it's going to be all drugs. So one thing we're often asked is, how do you prioritize? That's a lot of drugs, even though it's a minority. And we have listed the prioritization categories that we think about when we're deciding what CPIC guidelines to move to the top of the list, and internally at St. Jude, what we want to implement first. And of course, it relates to these questions. Is there prescribing actionability? The severity of the consequences of getting it wrong if you don't use genetics. Having a CPIC guideline makes it easier for us, but we don't always go by that. Is there an available clinical genetic test? How commonly used are the affected drugs? How commonly used are the variants? How common are the variants? Is there mention in drug labeling? And are there other professional prescribing recommendations that already exist? At St. Jude, we've been implementing pharmacogenetic testing since 2011, and we've enrolled 4,500 patients where we're preemptively testing patients. This is, again, the list of all the CPIC gene drug pairs, and highlighted in yellow are the ones that we've implemented at St. Jude. Notice that these last few have been ones for which we don't yet have CPIC guidelines, but they've been important to our patients. So we had a patient with a major GI bleed who was a CYP2C19 ultra-rapid metabolizer, and we really needed to take advantage of the Dutch guidelines to decide on the change of the dose of the proton pump inhibitor in that patient. So one of our original goals when we made CPIC was so that we could better understand what evidence is needed to decide to implement, and we think that we have gained a lot in this process of understanding what evidence is needed to move something into clinical actionability. I'd like to acknowledge my colleagues at CPIC, especially Terry Klein, and I think Michelle is here in the audience, and I think Serene is here too, as well as members of our CPIC SAB, some of whom are here, and our steering committee, and our colleagues at St. Jude in the PGen for Kids protocol. And I'll stop there. Thank you. She's asking about the API for CPIC, which is definitely on the top of our to-do list. That will make it easier for people who want to sweep the website on a regular basis to download all the information. We are definitely interested in getting feedback from users of the CPIC site about exactly what kind of information you want. We can't guarantee that we can modify the way that we create the database and the API to accommodate that, but we certainly want to get as much input as possible as to what information is most useful. Currently, all the data that underlie these tables that link genotype to allele, that link alleles to diplotype, diplotypes to phenotypes, phenotypes to prescribing actionability to specific CDS for specific drugs. All of that lays in different tables, and what we have to do is integrate all that into an API that'll make it easier to integrate with EHRs.
Sure. So Mary, would you like to comment on the discrepancies that you often see between guidelines, especially not necessarily between CPIC and PharmGKB, but also between FDA and DOA?
Okay, so PharmGKB doesn't make prescribing guidelines, but you're right, there's others, like we've done a formal comparison between CPIC and the Dutch guidelines and identified just a few discrepancies. The biggest one related to just some arcane questions about CYP2D6, and that's led to a multidisciplinary international group trying to resolve those discrepancies. So We want to try to harmonize with other professional guidelines wherever possible. CPIC does not agree with all of the FDA information about pharmacogenetic testing that has made it into drug package inserts. So I doubt if we would agree with everything in the European agencies also, but I haven't formally compared that. You can look on PharmGKB, and they make a very useful site that will link you for every gene drug pair or to the regulatory agencies, EMA and FDA, as well as clinical guidelines from CPIC and the Dutch group. And I'm sure that there are some discrepancies, but of course we try to resolve those as we go through creating every guideline, and we make a very big effort with every guideline to have international authors. So especially when the gene variant is of higher frequency in certain populations, we try to make sure to have authors who have expertise in those populations that have the high risk allele frequency higher to be involved. We want them to be guidelines that can be used across borders, but that's probably not always possible.
Any other questions? Mary, one question. So the NUDT15 variant, besides St. Jude, where could you order the test?
So actually at St. Jude, you can order the test, but we don't perform it. We send our samples to a company called RPRD. They came out of Medical College of Wisconsin, and they're a CLIA lab that does the testing. I think there might be one or two other labs in the country that are doing it. And so it's a good place to look for companies that do clinical genetic testing is the GTR, the genetic testing resource. Hopefully they're listed there.
Okay, thanks so much.